Glutamate is a small molecule neurotransmitter with an amino acid structure. It is the most excitatory neurotransmitter in the body. Neurons can terminate at a muscle gland or another neuron. Glutamate is a neurotransmitter released from axon terminal of the presynaptic neuron to the dendrites of the postsynaptic neuron. This transmits an electrical message along a nerve pathway. This junction between two neurons is referred to as a synapse. The action potential travels to the axon terminal of the presynaptic neuron. This causes the voltage-gated calcium channels in the synaptic knob to open. Calcium ions enter through the open channels. Vesicles containing glutamate move towards and fuse with the membrane of the synaptic knob. Glutamate is released into the synaptic cleft by exocytosis and binds to the receptors on the postsynaptic neuron. The chemically gated ion channels on the subsynaptic membrane of the postsynaptic neuron open. Cations enter the cell. The overall charge within the cell becomes less negative. This is known as depolarization. This is an excitatory synapse, as it brings the membrane potential of the postsynaptic neuron closer to threshold potential. Three important receptors that glutamate bind to are kainate, AMPA, and NMDA. NMDA and AMPA are multimeric ligand-gated ion channels. When glutamate binds to these receptors, ligand-gated ion channels open. However, the magnesium block on the NMDA receptor prevents the calcium ions from passing through the NMDA channel. Sodium ions enter through the open AMPA channels, resulting in the depolarization of the postsynaptic neuron. This releases the magnesium block from the NMDA receptor. Calcium ions can now pass through the NMDA receptor, allowing the calcium secondary messenger pathway to occur, which allows the addition of other AMPA receptors, increasing the neuron's sensitivity to glutamate. Excitatory amino acid transporters remove glutamate from the synapse into glial cells. Glutamate is then converted to glutamine via the enzyme glutamine synthetase. Glutamine can then be transported back into the presynaptic cell, where it can be converted back into glutamate for synaptic transmission. This process is essential as it protects the brain from excitotoxicity by glutamate.